So here we go. It's back. So interphase we said was 2n equals 2. 2n equals 2 means that there's one chromosome with two sets of genes, right? One from mom, one from dad. Here color-coded, mom will be green, dad will be yellow. And each gene is located in the same spot of each chromosome. So here might be gene A, and there is the homologous copy of gene A. Now remember, it does not have to be the same as the copy that came from mom. Dad may have provided a different version of that gene. So here we'll say dad provided lowercase a. What's the name for different versions of a gene? There we go. Oops, I added an extra E on allele. Let me get the eraser. And a gene is just a sequence of DNA that codes for a protein, right? Now, just so you know that what we talk about, when we talk about dominant and recessive, what we're actually talking about are the simplest human genes, or any genes, to study. A little bit of background about this before we go any further. Some of you wrote the name Gregor Mendel on your what you knew about genetics. Who is Gregor Mendel?
He is the guy who was a monk, and he crossed bean plants to make a different bean plant out of two of the same bean plants with different shapes.
So, absolutely correct. Gregor Mendel was an Austrian monk, and living in a monastery, you get assigned certain jobs. His specialty, his background was science. It's what he was trained in before he went to monastery. And monasteries, whether or not you realize it, are self-sustaining. That means that they have to do something to earn money to pay for all of the financial needs of the monastery, and they do their best to keep that as inexpensive as possible. And to keep it as inexpensive as possible, they do a lot of their own gardening to grow food. So somebody with a good background in science and botany would likely be assigned to working in the monastic garden. And one of the things that they would grow would be high-yield plants, so something that makes a lot of food, and a lot of food that can be dried out and used later in winter. Vienna and Austria, they have winter, which means they don't have a 24 or a 12-month growing season. It's just like us, where they have that window of growing in the summer. So their job then would be to plan ahead and think about that for things that could be dried out and preserved. Peas. Peas, very nutritious, very easy to dry out, and you can cook with them all year long then. They'll last you until the next summer when you can get fresh food again. So Gregor Mendel, part of his work was in that monastic garden, and he noticed that pea plants, when they were growing, had multiple traits, of which there were only two varieties. Pea plants were either tall or short. Their flowers were either terminal, so on the ends of the branches, or axial, which basically, instead of thinking about the ends of the branches, think of it like if I'm a branch of a plant and this is the main stem, and these are my shoots that grow. Axial would be like at the armpit of where the branch and the shoot meet. So they were either at the tips or the armpits. The peas were either green or yellow. They were either round or wrinkled. Their pods were either fat or they were what we call constricted, which means you could see the pea inside of the pea, like the physical pea inside of the pod. So they were constricted like that. And there were only ever two choices. That's it. There was no medium. It was tall or short. There's no in between. There was no like weird hybrid between yellow and green pea plants, which is good because that color would look like baby poop. Think about it. Blend the two. there's only 2 choices. And that's because only 2 alleles existed for each of those traits. So the only 2 versions of a gene, of each of those genes you could get, were the traits that he saw. Now, here's the problem for Gregor Mendel. We call him the father of genetics. And we call him the father of genetics because he's the guy who came up with this idea. But he was working in the 1800s, late 1800s, pre any of our knowledge about DNA. We didn't know how things were transmitted. We just knew that they were. So he had no explanation for his observations. And when I say his observations, I mean he, God bless him, this is how you know he was a holy man, peas naturally self-fertilize, so they don't have to have a pollinator. They'll just fertilize themselves, and if a tall plant fertilizes itself, it's going to produce another tall plant, right? Gregor Mendel was like, I have an idea. I know how this pollination thing works. I'm going to take this paintbrush, and I'm going to pollinate this tall pea with pollen from this short pea. 1 flower at a time. How many of you are like, yeah, if he did 10 flowers, no big deal. He did hundreds of flowers and hundreds of trials.
He has some patience.
Right? Holy man. The only person on the planet I can think of who would be patient enough to do that would be somebody who's clearly praying at the same time. Like every flower was a Hail Mary, because I would have given up a long time ago. A rosary was long enough for me. So, God bless Gregor Mendel. And he comes out and he publishes his results. He's like, hey, world, we call them character. You get these peas, they pass a character on from the parent pea plant to the offspring pea plant. And he discovered that if you took the tall plant and pollinated it with the short plant, you didn't get a medium plant like everyone would expect. You got tall plants. And if you pollinated a short plant with a tall plant, you got tall plants. Short plants disappeared in that generation. He's like, this doesn't make any sense. So then he let that next generation self-pollinate. I personally think that's where he gave up. He was like, dumbest idea ever. Should never have started this. I'm never going to end it. I'm done. So he let him self-fertilize. And because, remember, how do you get more peas to eat? You plant the peas that you dried out from the previous season. So he took those dried peas, he planted them again, and in his next generation, it was a strange combination between tall and short when they were allowed to self-fertilize. In fact, it was 75% tall, 25% short. And Gregor Mendel was a brilliant man. He's also a teacher, a biology teacher. One of the other monastic jobs he got. And he's like, you know what? This ratio keeps happening over and over and over again. This isn't random. There's something here. There's a reason for it. So he wrote it all up and he published it and everyone laughed at him. They rejected his idea. So much so that this is how you know while he was a holy man, he was also human. He gave up all of his scientific research. He was like, fine, world. If you don't like it, I am just going to do administration at the monastery. So he basically became a rector. Think of it like house mom for the monastery. That was his new job because his science was rejected and he poured countless thousands of hours into it. So he's like, fine. And it wasn't until, the 1920s and 1930s that we revisited this idea after Gregor Mendel. Poor Gregor Mendel. So I make sure we give him some credit every time we get here, because that's a lot of life's work to have somebody look at you and be like, no. It's not even like it was a dumb idea. It's not like he was like Lamarck. At least he said something that had statistical value. No? Question? Comment? Concern?
I was going to ask, when you said that it was 75 tall, 25 short, was that originally for 250?
It was originally from his first filial or his second filial.
First,
First filial, it was originally 100% tall. And that's what he had to notice, how it went from tall that self-fertilize and make 100% tall, short that self-fertilize and make 100% short, cross-pollinate and you still get 100% tall, and then self-fertilize and we get 75% tall, 25% short. So he's the guy who is like, I know my statistics. That's why we like statistics. That's why we encourage you guys to use statistics. Because without the ability to do statistics and mathematical analysis, Gregor Mendel never would have ended up saying, we have these characters, and some characters are dominant, and some characters are recessive. And you get a character from each parent, and they come together, and if you get the dominant character, you only see that trait.
So in this case, tall was the dominant trait and short was the recessive?
You got it. So we can use Punnett squares. He's the guy. You can thank him for those. We can use Punnett squares to find Mendel's ratios. Now I'm convinced you probably have all heard some of that story before, right? Some of you guys have used that Gregor Mendel stuff in your things. Those of you who haven't, don't worry, I caught you up. That's the story, the highlights. Really, it's just that was like Cliff's Notes. And he's the guy then who said the words allele. He didn't say gene, he said character. It wasn't until later that we got the word gene. But there's a whole branch of genetics, this dominant recessive thing that we now name for him. We call it Mendelian genetics. So anything where there are only two alleles and one is dominant and one is recessive is called Mendelian. That's our nod to Gregor Mendel. Now, if Gregor Mendel only knew where genetics would be in 100 years of his publication, that's crazy.
here's what I have his publication.
Yes. It's not the most fascinating read in the world. Mostly because now it's common knowledge. Like, nobody really needs to read Gregor Mendel to understand the dominant recessive thing, because it's in every biology text everywhere. Poor Gregor Mendel. Now, I will say it's slightly more complicated than Mendel. Mendelian genetics is the simplest version, that's why we start there. But it turns out that there are actually genes that have more than two alleles. So you can't really make a pretty Punnett square for those. The probabilities become much more complicated. So I want to look at that gene that we'd look at so you see exactly what I mean. So this is an X chromosome of a human. And what we do as geneticists, as scientists, we take that chromosome, it's always shaped like this. This is called the long arm. Can you imagine why? The centromere would occur right here at this little indent on the sides. Long arm of chromosome X. Short arm of chromosome X. We didn't do any trickery or reverse naming. Any chromosome, the longest one is called the long arm. And here where it's banded represents where there are multiple genes. Now this is just the X chromosome. Lady, you have two of these. Gentlemen, you only have one. And these are the diseases associated with the human X chromosome. So if you get a mutated or bad allele at any of those stripes, you get a disease. For example, Fragile X syndrome, albinism deafness syndrome, immunodeficiency syndrome, Kennedy disease, Fabry disease, let's see what else is on here, Duchenne muscular dystrophy, Becker muscular dystrophy.
Thanks, Michael. It's all your fault.
Let's see. There's so many. Oh, that one's really not very good. I mean, a lot of these are really not very good, but that one's very bad.
What is it?
Lesch-Nyhan syndrome, you can't digest a certain amino acid, so you pee crystals. It's very painful.
Oh.
Most of those people die like 3 years. Like that's not. You're not looking at like a long life expectancy. When you pee crystals, you could imagine long life expectancy is.
It's not really.
You want to pee solid? Stabby solid?
Like pure.
Some of you are like, stop saying.
I kind of think it'd be like passing like a kidney stone or something like that.
Yes, every time you pee.
I would stop peeing. I would
Just
just.
have like eat that one piece. What did you say? That's messed up.
Other things like cleft palate. You guys know what the cleft palate is?
It sounds familiar.
So your palate is the roof of your mouth.
Oh.
And when babies are born, so when a baby is in utero, they develop symmetrically, and then the two sides come together and fuse. So the top of your mouth came together in two sides, and then they met in the middle and fused before you were born. Not completely solid, because that's how your head squishes to get you out, because it's got to, like collapse a little bit, and then you come back out, and it like re-pops open and then hardens.
Wow.
If it hardened before you came out, you wouldn't have come out.
If your little bump is right here.
Not on your soft tissue, it's your hard tissue.
Philtrum.
Yep, that is your philtrum. So, that, if it doesn't fuse, if it has a hole in it, the two bones don't come together, and you have a hole right here, all the way back. All the way back. Now, cleft palate is really bad because you can imagine you can't eat. Especially, mind you, this is how infants are born. And how do infants eat?
Like a drink.
Bottle, right? It requires suction. They have to be able to seal their mouths shut. You don't have suction with a cleft palate, so they can't eat. Really big deal. It's really, really very bad. Now we have surgeries now that can fix it. And those individuals are rare. It's few and far between. And we can fix it now. We can feed them with other methods. We can feed them directly into their stomach until that surgery fixes them. But that's kind of a big deal. You're like, whoa, that's not a good thing to inherit.
How do like scientists or like people, how do they know, how did they figure this out without
So-
causing mutations and like,
There are a few ways that we used to do this, and there are some modern ways of doing this. Essentially, what we used to do is actually look at the protein and then take the protein product that gets made and then basically unmake it, like reverse engineer it to find the recipe for it, and then locate the recipe in the recipe book. Took a lot of work. By a lot of work, I mean tens of millions of dollars. Now we can do it with a different method called CRISPR. Basically, look at it this way. We take the particular chromosome in question, we cut it up with a bunch of chemicals, and we read them little bits at a time, and then we figure out what the little bits say. We put the whole recipe together to figure out what the recipe makes. It's faster, it's cheaper, it's really fascinating, and we're not even a tenth of the way into the human genome. Like, there's a ton of stuff that we have no idea what it does. And now we're discovering that there's a ton of stuff up here that's labeled as one thing that actually does like 4. We're complicated.
You said like the child's had indents of it at birth and then it hardens, but it was actually more. If a doctor was to accidentally apply pressure to any part of the head, would the child be born with like an indent around the head? Or
That will kill the child.
that would kill the child.
So, you ever held a baby?
I am not allowed to hold a kid. I'm not kidding right now. I
do you have a little brother?
almost dropped one. And it wasn't my fault. It was not my fault. The mother, I was giving it back to the mother, and then she prematurely grabbed it, and then it fell out of my hands.
Well,
She caught it, though. She caught it. Remember me.
okay.
It wasn't a newborn or anything. It was
So-
it was a 2-year-old, but I mean.
I will give you a little primer on the whole of the holding children thing. Any person, not just the doctor, who holds a newborn through probably 3 months, you have to support their little heads and you cup it with your hand like that. And you can't squeeze their little heads because their little heads are squishy still, and you can see the soft spots. Yeah. Their brain is in there, and if you squish their skulls, it's not that it won't pop back out after the squishing, it's that you squish their skull into their brain. And that's really, that's bad. We don't want to do that. Yes, they can use, they look like salad tongs, they go around the baby's skull, and they just pull it out. But they only use those if they're absolutely desperate and the kid gets stuck.
Oh yeah, that was me. I had a huge head.
They avoid that at pretty much all costs. So much so that instead of using those, they would rather cut the mother open than use the salad tongs. Oh no,
No.
if the kid gets stuck there, they're cutting the other part.
Oh no! No.
You mean when I talk to you guys about where babies come from, all I had to do was say that, and you'd be like,
You said, the guys, you told us being crystals, you told us that if we went to this one river in this one place that's not
I don't
here,
want to be
that
cut
well, friends, this is all of the glamour of being an adult.
we should burn the earth and move to Mars.
We would still reproduce the same way.
No, like, get rid of all the mean things here. Put all the mean people here.
One, you just suggested burning people. That's immoral.
Would you put them in a space bunker?
No, that's not going to work. So, all of these particular diseases here, most of these disease-causing genes in the X chromosome are Mendelian. So there's a good copy and there's a bad copy. Some of the bad copies, the disease copy alleles, are dominant. Huntington's disease, that's one, that it's a dominant disease. You get one bad copy, you've got Huntington's. There's no way around that.
What is Huntington's?
It is a muscle decay disorder that onsets. Early onset is at the age of 40, late onset is around the age of 60. Some onset is, early onset is as early as teen years. You go from being what looks like a perfectly healthy and normal adult to someone whose muscles decay. You can no longer walk, you can no longer talk, you can no longer eat, you lose brain function. It's a very unpleasant decline. That's dominant, so one bad copy. That's all she wrote.
Can we fix that?
Nope.
What if like you cross and then?
One, it's not possible. Two, there are scientists who are currently attempting to fix genes in utero. It's exceptionally immoral, and they totally kind of just got scolded by the rest of the scientific community for doing that. Yeah, they got yelled at.
What if you like?
They don't have any friends
What if
anymore.
you make like a fake baby copy?
What if you what?
A fake baby copy. You practice on that.
That's what they did, and they got in a lot of
No,
trouble.
not like an actual baby. It's like a model baby.
Right, it's like you can't.
Yeah, like a plexi baby, you know.
Why do you have to put stuff in it?
Yes, the only way it's going to help a human is if you actually change the human. And then, in which case, it introduces a lot of moral questions that we might not want to go into.
Plastic baby.
I could buy a doll.
Even if you were to not practice, like if you were to practice on a plastic baby and then transfer that knowledge to a real human, you would still have to do some really immoral stuff to make it transfer to the human.
Alright, this might seem like out there, but you know like the Spider-Man where like they cross with like the lizard, you know, is that possible for humans though?
No.
Do you want to be Spider-Man?
No, he's talking about the lizard. Yeah.
So
I didn't watch those movies.
here's the thing about this particular chromosome. Ladies, you got 2 copies, right? Yours are located, like your Fragile X syndrome gene is located in the same spot on both of your copies. So they'll be right next to each other if you lined up your second X chromosome. Sorry guys, you got Y. Y is about that big in comparison to X.
We're cool.
And that means any of these disease-causing genes, you only got 1 copy of it. So you better hope that mom gave you a good one, because you only get 1 shot in that game. And if you got the bad one, that's the one you see.
Well, who girls are good?
Wait, so does that mean that these diseases are most prevalent in children?
They mean they're most prevalent in men.
Oh.
Wait.
Because you only have one
You only get one X, so you only get one version of that allele. And since you only get one version of that allele, if the one that mom gave you was bad, then you have the disease. That's rough.
With girls though, like, wouldn't you have more chances to get the disease?
The vast majority of these diseases, while some of them are dominant, most of them are recessive, so she'd have to get 2 bad copies. And getting 2 bad copies is significantly less likely than getting a good one. That's rough. It's rough.
Now, let's say your kid is going to be born girl, and you know before the birth. Is there a way like scientists could, poke around and take away chromosomes and like that, and make
Change it?
a boy? Yeah.
No.
What did she just talk about?
I do want to emphasize to you this whole fertilization thing. So yesterday I made you make gametes, right? Sperm and egg. When do you get your full complement of genes? When does an individual get every gene it will ever have? At fertilization, right? So when that egg and that sperm meet, this individual now has every last bit of DNA. Once these two add together, every bit of DNA they will ever have. If you are going to make a genetic modification that every cell of this person will have, you have to make it right now. At the moment of conception. Because if you do it at any other time, you won't be able to change every cell in the body. The only time you can change every cell in the body is if you change the first cell that makes every other cell. So no, that's not possible. Does that answer your question with the biological foundation?
I have a follow-up, but I don't think it's going to really help.
Great.
I'm not going there.
So this moment, fertilization, that moment of fertilization we call what? Fertilization, it's called conception, right? The moment of conception, when those 2 chromosomes come together, we now have 2 individual cells that were n equals 1, who have fused to make one cell that is now 2n equals 2. So we have a full complement of DNA. 2 copies is all you're ever going to get, and that's it. What you get is entirely dependent upon meiosis in each of your parents. That's why we did meiosis yesterday, to show you where and how those genes separate in mom and dad, that you did the great big shuffle of half mom, half dad came together, made you. You sometimes see more of mom or you see more of dad because of which genes are dominant, which genes are expressed, where they're located, whether it's on the X, whether it's on the Y, whether it's on an autosome. Remember 1 through 22 that don't affect gender? So that's how you end up as you. This whole thing is kind of a big deal. You keep smiling like you're really just dying to know your question.
I know what's like, I feel like I know what you're going to tell me, but
We're just asking.
Oh no, because parents would love to hear this, I'm sure. Fire away!
that's also like, why I'm not asking it.
Just ask the question.
Just go for it.
If there were a scientist there at conception, and they and they altered the sperm before fertilization, it's not my problem either, because it's just if they were, and they were to alter the sperm before fertilization, would it be possible? Is what I'm asking. I'm asking like, it has somebody trying to ask if it would be possible.
That would be possible only through in vitro fertilization. That being possible only through in vitro fertilization is what some of those scientists attempted to do that they got ostracized from the scientific community for. The United States, the National Institutes of Health, has come out in a statement. Their director, Francis Collins, has said, we in the United States will have no part of this. It is immoral. It is unethical. It has consequences that we cannot predict. So the researchers who got ostracized are not from the United States. They're from a place whose government did not make that kind of ethical stand.
China.
And yes, and right now they're dealing with the backlash of the rest of the world
If you're
because
ostracized from-
that's, bad.
From the scientific community, like, I'm not saying what's happened, but like, if you were ostracized, like, you as a biology teacher, if you were ostracized from the science community, would that mean, like, they'd strip you of your, like, biology teaching ability?
It means it'd be really hard for me to find a job.
Okay.
That will follow you everywhere.
Now, like, when they ostracize them from the scientific community, they're from another country, would that ostracize them from, like, their own?
Every country.
Okay.
Well, like,
That means if those researchers attempt to leave China, they will not find a job. They will not find a university that's willing to take them. They're donezo.
Usually
That's
when
it.
you change your name.
So,
it's like harder consequences. Like, the whole world knows about that. It's like if they you did something, maybe Michigan would just know about that.
the thing is, the technology needed to do this would cost millions of dollars. So the only place that this is being done is at university, or could be done, is at universities or hospitals. You have to pass a review board who's going to say whether or not you can do your experiment or not. That kind of experimentation doesn't make it past a review board. When I say Francis Collins said we're not doing it in the United States, when he issued that statement publicly, he wrote the letter, he published it with his signature, with his title. He said, not only are we not doing it in the US, we're calling for every other researcher in every other country. We need to come and meet and make an agreement about whether or not this is going to be okay. Because we're there. We're at the point that we scientifically, we can do it. It's the question of should we do it that scientists are currently grappling with. That's a much bigger, much more difficult question than the how do we do it. Which I know you're like, wow, the how do we do it seems really hard. The should we is much harder. You know, I always tell you that theology is harder than biology. That's one of those things.
But I mean, what if you could like make a world without disease?
So you're saying every person who's born with a disease right now isn't worth living?
No.
Okay.
I'm not saying that.
But isn't that what that assumption says?
No.
Aren't you
I'm
saying
just
I'm,
saying-
I'm trying to get rid of all disease? So hey,
No.
you with a disease, I think you shouldn't have been here.
I just think everyone deserves a world
Objection,
without
Patrick,
disease.
veteran witness.
I'm just introducing questions of philosophical and theological importance.
So this is how the argument would go between scientists. Like, I would say the world without a disease, and you would say, what's wrong with disease?
No, that's actually not what it would sound like. We would start having a modern worldview versus sacramental worldview, nature as plastic, nature as sacred argument. Very different terms, but kind of. I made it for freshmen. You don't really draw a winner by saying that people who currently exist should be fixed. The last guy who did that, he kind of died.
Hitler?
What?
Yeah.
Well,
Well, his scientists also died. Mengele did not meet a good end.
they almost caught one of the scientists after death, purposely just stab people and took off their legs and stuff.
Who's that?
Horrible things happen. Horrible, horrible things happen. All because, mind you, your DNA has serious consequences on who you become, right? It all started with Gregor Mendel. Oh, the irony. It was a monk who brought up all of this how inheritance happens thing. And now he, in his foundation in science, is leading us to debate whether or not we should be doing things to human beings that would violate Catholic theological thinking. So I guess it's kind of an ugly reality that we have to face. And part of the reason that we talk about this stuff, even though it's not the stuff that's in your textbook, is because I think that this stuff is as important as the stuff in your textbook. Because you know who's going to be able to vote on whether or not we should be able to do this stuff in the future? Yeah. And I want to make sure that you leave my classroom understanding there are huge consequences, gigantic ones, to messing with the human genome. Ones that we can't foretell, ones we can't understand, ones we can't predict yet. Those scientists who have been ostracized actually had to write in their study that yes, they modified human genes, and it turns out, yeah, it kind of sort of did what they thought it was going to do, but it also had 10 other consequences they didn't predict that would have made the embryo, if it were allowed to develop into a human, it would have made it more sick than the original disease. Oops. Didn't see that coming. That's why Francis Collins is like, hey, hands off, because you don't know what you're doing yet. Big deal, huge deal, gigantic deal. And I want to make sure you leave me in a way that I can trust me to you when you're voting and making decisions and I'm living in the old folks' home.
You can
We
still
would
vote
never
on
let
it,
you
though.
go to old folks' home. We would put you here. We would put you here.
Why would you want to be here?
I'm just saying, I am fully aware who I have to trust the world to is you, who comes after me.
Hey.
They hurt people at old folks' homes. I mean,
Not all old folks' homes.
we should send in a spy dressed as an old folk.
Yeah, there's definitely so much bad activity going on in an old folks' home. We need to spy on those.
We just moved my grandma to an old folks' home with the nuns. I don't really think the nuns are, you know.
Well, that doesn't count. I'm talking about the one where like they're all greedy.
It is so creepy.
The nuns don't count. Not in the movie.
Muhammad based-
Nobody who's a good
It's like
person
Muhammad based
counts.
all of its research on movies. Although I did too on Spider-Man.
At least you're willing to admit it.
Yeah.
See, I thought it, but didn't say it. I was like, he'll catch himself.
Hopefully, you just have the type of person to watch movies.
So just so you know, there are some words that Mendel developed. Of course, they're based in Latin because, well, he was a monk. The words that you should know that kind of are associated with this is P is your parental generation. So whenever we talk about the two initial individuals that we crossbreed, those are your parents. And whenever we talk about the offspring of that generation, we call them the first filial. Filius, remember, son. So first filial. Second filial will be the offspring of the offspring. Now you're like, whoa, wait a minute, humans, offspring of offspring. Whoa, whoa. Don't worry, when we say offspring of offspring like that, we usually mean plants. It's socially acceptable for plants to, you know, interbreed. We don't judge them for that.
We can't. We
Poor
eat food
animals.
from them.
Animals surprisingly don't interbreed with siblings particularly often because they can smell the similarities in their immune systems and they know then, that they're related. So they intentionally seek another animal with a scent that is different than their immune system to avoid inbreeding. Isn't that cool?
Isn't that what King Tut did?
Most of the Egyptians, there's a significant amount of consanguinity, yes.
Uh,
Ugh.
if two animals from the same family were to breed, would they have the same consequences if two humans were to do the same?
Well, no, not for like the first 5 generations.
So if it continued on, then they would start seeing.
But it's same with humans. You wouldn't see the consequences until a few generations in of inbreeding. It's just, you know, socially, we frown upon that. So what Mendel saw when he initially watched his pea plants, he just said, if I allow a tall pea plant to interbreed with a short pea plant, and remember, he knew nothing about meiosis, so he didn't know how these characters got divided out into cells. All these are is representations up at the top of this Punnett square. It's just a representation of the 2 alleles that tall parent would have made. 2 tall alleles. And on the side, it's 2 short alleles, the representation of the 2 alleles that the short parent could have made. Now, we cross them in the box, remembering always write tall first. Dominant uppercase letter always goes first. Lowercase letters always second. We cross those and we see the only potential combination of offspring that are made is 1 capital T, 1 lowercase t. There's no other options. All of those are going to be tall because capital T is dominant to lowercase t. So we have 100% tall first filial. Now, there's a few special names here. The genotype are the 2 alleles an individual possesses. So the genotype of parent 1 is capital T, capital T. The genotype of parent 2 is lowercase t, lowercase t. The genotype of the offspring 4 of 4 offspring are capital T, lowercase t, or heterozygous. So they are 4 of 4, capital T, lowercase t, or heterozygous. And it's genotype. But remember, Mendel had no way to see the genotype. There was no way he could look at chromosomes. There was no way he could look at genes. He didn't know what those things were. So even if he could have looked at them, he wouldn't have known where to look for them or what the heck he was even looking at. Instead, he could only report what he saw. The traits that you see when you look at an individual are called their phenotype. So the phenotype would be the trait that you see when you look at someone. That phenotype here would be 100% tall. That's what you see. That was his first test cross. So he took his self-fertilizers, he hand-fertilized them, made a test cross. Then remember I said he let this self-fertilize like it normally would. And we'll see why he got the results that he got. The only 2 alleles that individual could have made was a capital T, lowercase t, right? Or the only 2 gametes, because those are the only 2 alleles it had. So my genotype here would be 1 of 4 is homozygous dominant. Homozygous dominant or homozygous capital T, capital T. 2 of 4, and since fractions are our friends, 2 of 4 is actually
Fractions are wholes, I can't stand them.
1/2 of them are heterozygous, and then 1/4 of them are homozygous recessive. That's our genotype for this particular cross. Our phenotype for this cross would be 1, 2, 3, 3 of 4, or 75% tall, and 1 of 4, or 25% short. That's what Mendel saw.